The cell cycle begins with interphase, during which the cell is preparing to divide. The nucleus is clearly visible as a distinct organelle surrounded by a membrane. The chromosomes are not visible as distinct rod-like bodies. They appear as a long thread-like mass of material called chromatin. One or more nucleoli are usually visible. Following the G2 phase is mitosis, or the M phase. Mitosis is actually 4 phases. The first phase is prophase. During prophase, the nuclear membrane begins to break down. The nucleoli disappear. The centrosomes divides and begins to migrate to opposite ends of the cell. There are no centrosomes in plant cells. These structures produce the spindle fibers that attach to the centromeres of the chromosomes. Plants can produce spindle fibers without centrosomes. During prophase, the chromosomes become visible as 2 distinct chromatids joined by a centromere. Kinetochores, locations on either side of the centromere where the spindle fibers attach, are produced late in prophase. Metaphase follows prophase. During this phase, the mitotic spindle fibers attach to the kinetochores. The chromosomes begin to move towards the center of the cell. During this phase, the nuclear membrane completely disappears. During the next phase, metaphase, the chromosomes are oriented across the center of the cell by the spindle fibers. This is referred to as the Metaphase Plate. Here the chromatids begin to separate into 2 chromosomes during Anaphase. The centrioles produce the spindle fibers that attach to the kinetochores. Anaphase follows metaphase. This phase begins the separation of 2 complete sets of chromosomes. The 2 new single-stranded chromosomes begin to move away from each other towards opposite ends of the cell. In early anaphase, the 2 groups of chromosomes are a short distance apart. Later, they have almost reached their respective ends of the cell. In the final phase of mitosis, telophase, the 2 sets of chromosomes have reached opposite ends of the cell. The spindle fibers attached to each chromosome disappears, and a new nuclear membrane forms around each set, so each new daughter cell will have its own nucleus. The chromosomes resume their interphase form, long thread-like masses of material, chromatin. The nucleoli also reappear during telophase. Cytokinesis is the actual division of the cytoplasm. In animal cells, this can begin to be seen during anaphase and continues during telophase. Animal cells begin to be pinched apart by forming a cleavage furrow near the center of the dividing cell. Eventually the furrow continues until the new cells are separated. Cytokinesis in plant cells is different. A cell plate, which will separate the two cells, is produced in the cytoplasm between the new nuclei during telophase. This plate develops from the center of the cell towards the opposite sides cell walls. The end cell walls of the new cells are eventually formed from the cell plate. The two new cells produced will then enter interphase, and the cell cycle starts over again. DNA is produced during the S phase of interphase. During prophase, the chromosomes become visible and the nuclear membrane begins to disappear. Metaphase, the mitotic spindle fibers attached to the kinetochores, the chromosomes begin to move towards the center of the cell. The nuclear membrane completely disappears. During metaphase, the chromosomes move to and orient across the center of the cell. Anaphase, the sets of chromosomes separate and move towards opposite ends of the cell. Again, during the last phase of mitosis, telophase, cytokinesis occurs, producing a cell plate which divides the two new cells. The entire cell cycle, interphase, prophase, prometaphase, metaphase, anaphase, and telophase.